The next layer, next level. So we said primary structure is the sequence of amino acids in the polypeptide chain. And that's really, really important because mutations in that sequence can lead to protein disease states. Example we gave was sickle cell anemia. Secondary structure, alpha helix, beta sheath. We get this from hydrogen bonding between atoms of the backbone. So oxygens and hydrogens from the backbone can bond amino acids together. We get alpha helices, beta sheets. Tertiary structure is the way in which an entire protein chain is then coiled or folded onto itself. It gives a specific three-dimensional shape. So we take that alpha helix, or we take those beta sheets, and then we coil the rest of the protein. It looks just kind of like a wadded up piece of spaghetti, but it has purpose to it, and it is not random. The things that hold it together, so like what keeps it from just unraveling again, is there are interactions primarily between side chains. This is what denotes tertiary structure. So secondary structure was interactions between the backbone, protein backbone. Now we can have interactions between the side chains because they're going to be brought in close proximity. They're going to be twisted together, folded together. They're going to start to interact, and that's going to hold the protein into its functional shape. So these interactions can occur between two amino acids within the same peptide chain, forming an intra-chain loop, or between amino acids on completely different chains, bonding separate chains together. Okay, so you can get between the same chain or between two different chains. Sometimes proteins have multiple polypeptides like bonded together with hydrogen bonding or different interactions that create the protein overall. So an example is with insulin. Anybody know what insulin is useful for? What does insulin do?
It breaks down your sugars.
Do what?
It helps keep a steady blood sugar.
Regulates our blood sugar level, okay? Helps break down glycogen if we need a spike of glucose into our blood, okay? So this is an example. We have two chains here in insulin, and look, it tells you all the little three-letter, tells you identify each glucose here as like its own amino acid, okay? So we see the sequence in the polypeptide, and then we see this right here. Anybody, what is this? Why do we see this SS, okay? Well, it's telling us these are two different kinds of disulfide bonds. So between two amino acids, both, if you look at this, this is, what is CYS? Anybody remember what CYS is? What amino acid that stands for? They must be really important because these are the only amino acids involved in these intra-chain disulfide bonds, we're saying, or interchain. CYS, CYS. You look up at that chart that we looked at above, this is cysteine. Oops, CY, cysteine, okay? And cysteine has a thiol group, okay? So the side chain of cysteine, the alpha carbon, CH2, and we have an SH at the very, very end. Okay, so I think it's just one CH2. Is there another one? I don't think so. Cysteine, oh, right here. Yeah, just one. Okay, so when you bring these two cysteine atoms together, when they're in close proximity, they can form the disulfide bonds. They simply lose their hydrogen on the thiol, and then you can bond two different, the sulfur of two different cysteines together, and that's what you're seeing here. Here, it's happening within the same chain. Okay, so we call it an intra-chain disulfide bond. But we can also link this chain A to chain B. If we have cysteine amino acids on both chains, then we could get a disulfide bond between the two chains, and so we call it an interchain disulfide bond, and that's what you're seeing here. Okay, so this is what holds insulin together. This picture over here is just showing you these yellow. God, there's so many buttons here. If I accidentally hit one thing, it like jumps to a different screen. These yellow, these are the interchain or intra-chain disulfide bonds that hold insulin together. And it's like globular, the three-dimensional spherical structure. Okay, so that's tertiary structure, but disulfide bonds aren't the only kind. There's many, many different kinds of ways that amino acid side chains can interact. So we're going to hit all the ways now. Okay. First off, tertiary structure, we said, is interactions between the side chains. These interactions are non-covalent. What does that mean? Okay. Do you guys remember the difference between a covalent bond and an ionic bond? Anybody remember the difference? This is something you talk about early on in like the prerequisite chemistry course. Ionic bonds and covalent bonds. Anybody remember the difference? Ionic bonds are made up of plus and minus, separation of charge. You have cation and anion. An electron is totally given away or received by atoms. Covalent bonds, electrons are shared. Okay, so if these are not covalent bonds, these are non-covalent bonds, then the type of interactions between the side chains look a whole lot like ionic bonds. You're going to see partial positives, partial negatives, or full-on partial and negative charges, and that's going to attract side chains together. We know positive is attracted to negative. So one way this can happen, one way you could have non-covalent interactions are hydrogen bonds between R groups, okay, or between one R group, which is a side chain, and the backbone. So you could have two side chains interacting, or you can have the backbone interacting with the side chain. So let's look at this picture. This figure from your textbook shows all the different kinds. And here's one example, okay? You could have hydrogen bonds. This looks like it's hydrogen bonds between the backbone. Okay, because we don't see any side chains sticking out. So we know we can get hydrogen bonds just between the backbone, but another way we can get hydrogen bonds, look at this right here. Hydrogen bonds can occur if we have two side chains sticking out. Okay, so this right here, this is the side chain for serine. And this would be the side chain for, I think it's asparagine. All right, and you don't have to identify them, that's not important right now, but we have these two amino acids and their side chains are interacting with a hydrogen bond. Now, if it helps you, you can draw in the partial positive on that hydrogen and the partial negative to remind you this is a non-covalent interaction. You've got charges here. Now they're partial positives and partial negatives, but you still see those positive negative signs, and that's where the side chain interaction is coming from. So it's a hydrogen bond between side chains. That's one way. The next way you could have tertiary structure holding a protein together is salt bridges. These are ionic attractions between positively charged side chains and negatively charged side chains. So this is going to be, if you remember that table had three kind of divisions. You got the non-polar amino acids, the polar amino acids, and then the acidic amino acids. These are going to be those acidic, and some of them are basic technically, that always are carrying the positive and negative charges on their side chains. So if I could find an example of that, does it have a salt bridge anywhere? Yeah, right here. Okay, so this right here is lysine, and this right here is aspartate. And you see the full positive charge on the nitrogen group, the amine group of lysine, and then this carboxylate anion portion of aspartate has the negative charge. So the interaction here is the salt bridge. It's a full positive and a full negative charge. These are not partial positive, partial negative. You don't have to write the little Greek delta sign in. So this would be a salt bridge. Disulfide bridges. So we actually looked at the example of this with two cysteine atoms. Okay, and you see a picture of this down in the bottom right-hand corner. Okay, if we have two cysteine, so this has got to be cysteine. These are the only amino acids that can make disulfide bridges. Okay, methionine cannot because it doesn't have a thiol group. It has just a sulfide group. Okay, that sulfur in methionine is not terminal, it's internal, so it can't form disulfide bonds the way cysteine can. But two atoms of cysteine And notice, these cysteine atoms are not next door neighbor amino acids. These are like at totally different locations on the protein chain. And that's okay. That's very frequent for amino acids to interact and like be held together with tertiary structure. They can be at complete opposite ends of the protein chain. But when the protein folds together, it just so happens that they're next to each other, okay? And they form those interactions. Lastly, the last kind, hydrophobic interactions. These form between clusters of, notice, non-polar side chains. Well, I should circle the whole thing. Non-polar side chains that interact via van der Waals or London dispersion is usually the word you hear me say, forces. So those are highlighted in yellow. There's 2 pockets that we see here. And if you just notice, all the side chains here are non-polar. They're hydrocarbons. There's no oxygens and nitrogens and sulfurs here. They're just carbon hydrogen bonds. So we just have these non-polar groups interacting. You get all kinds of London, I'll abbreviate, London dispersion forces interacting with these side chains, holding things together, okay? These are all temporary. These are the weakest kind, but they do matter. You get another example over here, London dispersion forces with those hydrophobic side chains. Okay, questions here? Do y'all kind of grasp the difference between the different kind of non-covalent interactions? All right.
What did you say what the one on the very end was?
The one on the very end.
Is that anything or no?
Of the figure, Maggie?
Yeah.
Like, you talking about over here?
On the right.
Which part of the figure? When you say the very end, what do you mean?
Just the helical structure. Did you say anything about that or no?
Structure. This is showing these are hydrogen bonds between backbone atoms. Okay. Holding the helix together, or this is beta sheet. Okay, you see how it's a pleated sheet with these little contours right here and here and here, and it's 2 sheets lined up next to each other. Yeah, these are hydrogen bonds between backbone atoms that are holding beta sheets or helices together. We didn't talk about this as hydrogen bond between backbone atoms, but here's hydrogen bond between a side chain and a backbone atom, okay? So you can get hydrogen bond just between backbone atoms or between side chain. This is a tyrosine side chain interacting with the backbone, carbonyl carbon, or the oxygen on the carbonyl carbon of some other amino acid, okay? So, yeah, there's a lot of different ways that tertiary structure can play into holding a protein in its functional shape. Okay, now real quick before we move on to the last phase or stage of protein structure, I want to introduce the fact that you can also have non-protein parts be part of your protein for functional reasons. Okay, we call these conjugated proteins. So most protein, I'm not gonna say most, some proteins are simple proteins. And that means they just have amino acids only, okay? And you see all the different ways, alpha helix, beta sheet. You'll see beta sheets represented by a flat arrow, kind of like that. Loops and disulfide bonds, okay? You can get all of those together. But you can also then sometimes have conjugated proteins where there's one or more non-protein parts, like a monosaccharide or a lipid or an ion is part of your protein structure for a reason, okay? So these are proteins with one or more non-amino acid units incorporated in the structure. Example is a heme group. So hemoproteins, heme. We know heme is important for our red blood cells. This is how oxygen is transported. And heme, I'm just going to draw the basic structure. If this is heme does kind of have a square structure. If this is heme, right in the middle, you have an iron, Fe, okay? And it's like a +2 or +3, depending on what oxidation states it's in. And this is why heme can carry oxygen. And this is what gives red blood cells its red color, okay? Iron is typically red. You have a lot of iron content in red meats in our red blood cells, so it's a red color. And this is not an amino acid, right? It's just an ion. And so hemoglobin is a great example of a conjugated protein. But this whole table just gives you several different examples. So you can have glycoproteins. Now pay attention to the first part of the word, glyco, okay? The non-protein part would be a carbohydrate, all right? So glycoproteins, we talked about this with lipids. They're in cell membranes and they stick out and they have these monosaccharide parts that are important for receiving cell messages, signaling. But these glycoproteins, that's an example. The non-protein part is a carb. Lipoproteins, you could probably guess lipids are the non-protein part. You can have lipids tucked into proteins. So high density, the example is high density and low density lipoprotein. Anybody heard of HDL and LDL? The good and the bad cholesterol, okay? So this is an example of lipoproteins, and they have lipids incorporated with them, and they transport cholesterol or other lipids throughout the body. Metalloproteins, so metal ions tucked within the protein. Okay, hemoglobin is an example of that because it has the iron atom. So hemoglobin is technically a metalloprotein as well, but it's got its own category because it transports oxygen, and oxygen is not amino acid. Oxygen is a non-protein, but it's like a substrate for hemoglobin. Okay, but that's metalloproteins when you have metal ions, and the active site or part of the protein structure that helps it function. Phosphoproteins have phosphate groups. Hemoproteins have heme groups. Okay, these are important for, like I said, storing oxygen. And then you have nucleoproteins, which have different pieces of RNA tucked in there. So I just wanted to introduce this idea that you can have conjugated proteins and other pieces that are there for a reason for the protein to function. Okay.
Do you want us to know that for the test?
Yeah. Opt-in organ. This right here, I mentioned it earlier, but I'll hit it again. That consider the type of interactions. We have these hydrophilic polar interactions like salt bridges and hydrogen bonds, and then we have the non-polar interactions. Okay, when it comes to tertiary structure, and because our proteins are in the aqueous environment of our body, okay, which is mostly water, it will orient the hydrophobic interactions so that they're tucked on the inside. So like the non-polar side chains, those pockets of interactions will usually be on the inside because they don't like to be on the outside near the water. And then the hydrophilic interactions are usually on the outside because they can interact with water just fine. Okay, practice. So what type of non-covalent interaction occurs between the glutamine and threonine side chains? Draw the structures of these amino acids and show the interaction. Okay, so glutamine, I'm just going to draw the alpha carbon here. It's got its hydrogen bonded to it. And I'm going to draw the side chain for glutamine. So we got a CH2 and we got a CH2. So if you're wondering, how do I know what glutamine looks like? Go back and look at that table. I've got it right here on my laptop. And then this is the carbonyl carbon. And then we have an amide group is part of glutamine. Okay, so this is glutamine. And we have threonine as well. And if you remember, I'll draw the rest of glutamine though as well. Here's the carboxyl group and the amine group. Okay, so this is glutamine, GLN. Threonine, I'm going to orient it the other way. So I'm not going to draw it with the alpha carbon on the left and the side chain pointing to the right. I'm going to draw it with the side chain pointing to the left so that we can see how the side chains interact. I'm just trying to orient the side chains close to each other. So I'm going to draw the alpha carbon right here. That's the alpha carbon, the hydrogen. And so I'm kind of, drawing the mirror image. I'll orient it that way. And so this is the backbone of threonine. And then we've got CH2. And then we got OH. So I'm going to scoot threonine over so that it's kind of close. So THR is 3. Oh. Okay, so we got threonine it back over. Threonine and glutamine. Okay. And the parts that are highlighted, this is the side chain. This is the side chain of each of these. Okay. Highlighted in blue, we'll say this is the backbone. This is the backbone. Okay, so I've oriented them different ways just so I can get their side chains next to each other. So what kind of non-covalent interaction do you think we would have between these two side chains? Would we have, well, what's our options? Scroll back up. Would we have hydrogen bonds, salt bridges, disulfide bridges? Those are our three options because the hydrophobic interaction is, sorry. Hydrogen bonds, salt bridges, or hydrophobic. Disulfide bonds are covalent bonds. So hydrogen bonds, salt bridges, or hydrophobic interactions. What kind of interaction do you think we would have here? Hydrogen bonds, salt bridges, or hydrophobic interactions? Anybody have a guess?
Is it hydrogen?
Hydrogen bonds, because in black, we have this positively polarized hydrogen. Okay, we've got the lone pairs on oxygen. And guess what? This hydrogen on the OH group of threonine is also positively polarized. So these are both donors, and then the lone pair on the nitrogen here is another acceptor as well. So you can get all kinds of hydrogen bonds. This hydrogen is also positively polarized. So you could get the lone pair on nitrogen could interact with that hydrogen. Both of these hydrogens could interact with the oxygen. Okay, you could get all kinds of hydrogen bonds between the side chains. So hydrogen bonding. Okay, questions on that? I think this right here is a great thing you guys need to know, especially for the exam. If I gave you 2 amino acids and said, what kind of interaction would you get between these two? And I might not even draw them out for you, but if you could, you should be able to draw out the side chains and then consider what kind of interaction you would get. Okay, I think this is a great question too. Hydrogen bonds are important in stabilizing both secondary and tertiary structures of proteins. How do the groups that form hydrogen bonds in the secondary and tertiary structures differ? How do the groups that form hydrogen bonds in the secondary and tertiary structures different? So we can have hydrogen bonding. We know secondary protein structure is critical for hydrogen bonding. Or hydrogen bonding is critical, I said that backwards, for secondary structure of proteins. It's also useful for holding together proteins at the tertiary level. But how are the hydrogen bonds different? They're both hydrogen bonds, but between what groups?
Is it because tertiary can bond to the backbone?
It can, yeah, it can involve the backbone, but what else can it involve? Secondary structure, the hydrogen bonds there are only between backbone atoms. Okay, so for secondary structure, hydrogen bonds are only backbone atoms. Okay, and they're regular and repeating. They happen all throughout the backbone. So we'll just write regular. Repeating. For alpha helices, it happens every 4 amino acids, 3.6 to 4. For tertiary structure, hydrogen bonds, how are they different? Well, can occur between, I'm going to abbreviate, between side chains, and it can occur between backbone atoms. You could have hydrogen bonds just between 2 side chains. You could have hydrogen bonds between side chain and a backbone atom. You can have those different variations, and they are not repeating. They seem more random, but they're not as regular and repeating as the hydrogen bonds that hold the protein together at the secondary level. Okay, to give yourself a visual of this, if you're a visual person, go back to this figure and look at the pleated sheet structure. You see how regular and repeating those hydrogen bonds are? It looks like rungs on a ladder. Same idea over here with the helix structure. We know alpha helices and beta sheets, this is secondary structure. So these are backbone hydrogen bonds, and they're regular and they're repeating. But at the tertiary structure level, we get random hydrogen bonds here and here. They're not regular, they're not repeating. It can occur between backbone or side chain, or a mixture of both, okay? All right, a little more practice. Says look at table 18.3. That's the table, I believe, with all the amino acids. Is that table 18.3? I didn't put the table number, but it probably is. Identify the type of non-covalent interactions expected between the side chain of the following pairs of amino acids. Okay, so glutamine and serine, you get all these things, or all these pairs. So we're trying to look at the side chains of these amino acids and identify what kind of interaction would we get? Would we get a salt bridge? Would we get a disulfide bond? Would we get hydrophobic interactions or hydrogen bonding? So your four options are these four right here. 1, 2, 3, 4. You get some kind of interaction, one of these four. And I want to know what kind. So glutamine, we actually drew glutamine out, slide above, and serine. Serine is going to be just like threonine, because if you look at serine, serine has, I actually didn't even draw threonine quite right. I missed one part on threonine. We drew serine, we didn't draw threonine. I'm going to erase that too. It should be like this, CH3. That's threonine. Serine is what I did have drawn. So if the interaction, if the side chains are very similar, the interaction is probably similar, right? Between glutamine and serine. Should be the same kind of interaction you would get between a glutamine and a threonine. So you would get hydrogen bonding between the side chains of glutamine and serine, because the functional groups are just the same. Okay, what about B, isoleucine and proline? Isoleucine and proline. Look at the table, look at the side chains for isoleucine and proline. What do y'all think? Can y'all see the table? Do y'all have the slides? So isoleucine, this is the alpha carbon. The side chain is all hydrocarbon here. Proline is kind of weird. Proline, if this is the alpha carbon, and we have this amine group, kind of bonds the two together. But these are all carbons, hydrocarbons. So we have two side chains that are just carbon and hydrogen. Non-polar, hydrophobic. So I bet that means we would get hydrophobic interactions. Okay, so van der Waals or London dispersion forces. What do y'all think about C? Aspartate and lysine. Aspartate and lysine. What kind of amino acids are these?
Is it a salt bridge?
Yes, Abby. So aspartate has a negative charge associated with its side chain. Lysine has a positive charge associated with its side chain. So we get a salt bridge here. Okay. What do y'all think about alanine and phenylalanine?
Is it hydrophobic?
Hydrophobic. Okay, and if you notice, these names are similar, right? Alanine and phenylalanine. If you remember, a phenyl group is just the attachment of a, I'm trying to help you remember how these are shaped. Just the attachment of an aromatic ring. We call that a phenyl group. So alanine and phenylalanine are both hydrophobic. One just has an additional aromatic ring attached and is a lot bigger. So this would definitely be hydrophobic. So again, if you don't have this sheet or the table, those slides from earlier, showing you all the side chains of the amino acids, you're probably wondering how in the world are we coming up with the answers here? Be looking at those side chains, and then you're characterizing those interactions as one of these 4 different kinds of interactions from this slide here: hydrogen bond, salt bridges, disulfide, or hydrophobic.
Sorry, you probably already said this, but did you say we need to memorize the neutral side chains, or will you provide them on the test?
Y'all need to know all 20 side chains.
Okay.
I know, y'all looking at me like I'm crazy. But this is so, amino acids are so important. They're so important. Okay, you get a note card. I'm giving you a note card. So if you want to use a note card for your side chains, do it. But you need to have all 20 either in your head or somewhere, okay? All right, I'll let you do this practice problem, and this practice problem, I think these are great to practice on your own. We're gonna move on the last couple slides here: quaternary structure, and then things that can denature proteins. And then we're gonna get to exam 2. So quaternary structure, this is the last layer of structure in a protein, okay? So quaternary structure, a 4 with a degree sign is quaternary. This is the way in which 2 or more protein chains aggregate or form large ordered structures. So basically everything we've been talking about is within a protein chain. When you get multiple chains, like big long chains, and they start to link together into these different units. So a great example is with hemoglobin here. Can y'all see the different colors, like the teal, the yellow, the purple, and the green? Each of those is its own protein chain. So within hemoglobin, we have 4 chains linked together to form the overall structure of hemoglobin, okay? And that's the quaternary structure of hemoglobin, okay? You have these different subunits, each chain, or sometimes it's a combination of chains that can form subunits, and these subunits are linked together to get this huge globular or fibril type structure. Collagen is another example. For collagen, so collagen is really important for like our bones and our joints, and several different tissues. people who are low on collagen, they can have a lot of like joint pain, back pain. But what collagen looks like is this alpha helix. And what happens is this alpha helix, so this is a single alpha helix, and then they pack together like a braid. Okay, think of like a braid is 3 strands braided together. You get 3 alpha helices kind of packed together. They're not necessarily braided, but they're all together. So this is 3 strands, or 3, I should say, helices stranded together. And then the quaternary structure, you start to overlap and overlay these braided or coiled strands of 3. And then they overlay together to get this really tough fibril. Okay. That would be like the quaternary structure. This would be the quaternary structure of collagen. This would be the black picture above is the quaternary structure of hemoglobin. So specifically, I didn't say with hemoglobin, but it's composed of 4 polypeptide chains. 2 are known as the alpha chains and 2 are known as the beta chains, and then they're held together by hydrophobic interactions. Each chain has about 140 amino acids, and each chain contains its own heme group. So this is a heme group. This is what I drew earlier, just the square with the iron in the middle. This is a better, more accurate picture of heme. And each chain, so the yellow, the teal, the green, and the purple, each hold a heme group. And each chain can bind a molecule of oxygen. So each protein, hemoglobin protein, can bind 4 molecules of oxygen. Okay, so those are just examples. Let's do a little summary. So to give you the overall summary, primary sequence is the amino acids and their sequence, their order. It's primary structure. Secondary structure is the alpha helix and the beta sheet. Those are held together by hydrogen bonding. Tertiary structure is the folding of a protein chain. You can have a combination of alpha helix, beta sheet, but it's held together by non-covalent interactions, the salt bridges, the hydrophobic interactions, the hydrogen bonding between side chains. And then once you begin to join protein chains and you have multiple subunits, multiple chains joined together, that's quaternary structure. So that's primary, secondary, tertiary, quaternary. Okay. This is just a summary. I just really grabbed this from your textbook. It's just a summary of the different classes of protein. I'm trying to see if there's anything on there I want to mention specifically. There's really some words here like mobile proteins and native proteins that I haven't talked about. I will mention, the only thing I'm going to mention here is this term native protein, because what we're about to talk about. The native protein, so when a protein is folded into its accurate shape and its functional shape in the body, we call that the native protein. But if it's mutated, or if it's damaged, or if it starts to denature, then we say it's no longer in its native state. Okay, so if I were to mention like what is a native protein, or say the native protein hemoglobin, blah, 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 just know that that's in its functional accurate shape. Now we can begin to mess with proteins different ways. We can break them apart, we can unravel them, and that's these last, I think it's just last 2 slides is talking about what happens when we hydrolyze or denature proteins. Okay, so chemical properties of proteins. The first one to mention here is protein hydrolysis. So this was a multiple choice question on exam 2. Hydrolysis, okay? We take water and we break molecules apart. We call that hydrolysis. And the reason I put that on the exam is because hydrolysis is not going away. We can hydrolyze any biological molecule, but it is the breaking here of specifically peptide bonds within the protein to yield individual amino acids. All right. So there are enzymes that specifically do this, like they clip protein peptide bonds, and these are called proteases. Now, specifically, you see this word endoprotease, but these are enzymes that are proteases. And what they do is they clip the peptide bond right here. They clip it right there and they just break that carbon and nitrogen back apart. Okay, so if you see the word protease, you know what it does. It clips peptide bonds, breaks amino acids into their individual structures. Okay, 2 examples of proteases here. Trypsin is one. It specifically cleaves, it doesn't cleave every peptide bond. It only cleaves peptide bonds on the carboxy terminal side of lysine and arginine. What that means is on the right side. If it's written with the amino side on the left, carboxy side on the right. Okay, so this should be the carboxy terminal. This should be the amino terminal. So if we have a lysine or an arginine, those are the ones that have side chains with a positive charge on them. Then it's going to clip on the carboxy terminal side of that side chain, the peptide bond. It clips the peptide bond. And chymotrypsin is another protease. It cleaves peptide bonds on the carboxy terminal side of aromatic side chains. So your phenylalanines, your tyrosines, your tryptophan, anything with a aromatic ring on the side chain, it will clip on the right side of that amino acid. The peptide bond is what it clips. Okay, so this figure is just showing you the individual amino acids that you get when you clip this peptide apart. You can do this with really strong acid, so that's why it shows you. You can do this with H3O+, that's just indicating really strong acidic conditions can do this, or digestive enzymes, proteases. So let's put this into practice. What fragments would be produced when chymotrypsin acts on the following peptide sequence? So remember, it's not going to clip every single peptide bond. It's only going to clip certain peptide bonds. And chymotrypsin cleaves on the carboxy terminal side on the right side of aromatic amino acids. So which ones here are aromatic? ASP is aspartic acid or aspartate. Is that one aromatic? Do y'all remember? I know you just learned these today. No, this one's not. So yeah, Cameron said it. This would have a negative charge. So that one's not aromatic. What about tyrosine? Anybody remember if tyrosine is aromatic? Yeah, Claire's nodding her head. This one is aromatic. So I'll put a star there. So chymotrypsin will cleave right here. Okay, what about phenylalanine? PHE is phenylalanine. Phenylalanine is aromatic. It has, I think we talked about it earlier, right up here. Did I draw it? Okay, phenylalanine has this aromatic ring. It's a CH2 group, then an aromatic ring on the side chain. So that one is aromatic. So guess what? It will clip again right here. Glutamine, asparagine, cysteine, proline, lysine. None of those are aromatic. So these are the only 2 places chymotrypsin would cleave this sequence. So now write the fragments. I would get ASPTYR, that's fragment number 1. Then I would get PHE by itself, we call that fragment number 2. And then I would get the rest of the sequence. GLU, glutamine, or sorry, GLU is glutamic acid. I wrote GLN. Sometimes my N's and my U's can look alike. Oops. GLU, so glutamic acid, then asparagine, cysteine, proline, then lysine, then glycine. And I need to shrink this down. Okay, so this would be fragment number 3. Okay. Second question, what fragment would be produced when trypsin acts on the following peptide sequence? So trypsin, remember, cleaves on the carboxy terminal side of lysine and arginine. So you're just looking for LYS and ARG. LYS and ARG, and you're going to cut it on the right side, the carboxy terminal side. So this would be the N terminal. This would be the carboxy terminal on the right. So here's a lysine, it'll clip right after lysine. Here's an arginine, it'll clip right after arginine. I think that's the only places. Okay, so fragment 1 would be ALAPHELYS. And then we get CYSGLYASPARG. That'd be fragment number 2. Fragment number 3, so LEU, phenylalanine, glycine. And I'm just going to write an A for alanine because I didn't leave enough space to write all 3 letters. It'd be fragment number 3. Okay. All right. Last slide. Questions on how trypsin and chymotrypsin work. What they do. They cleave peptide bonds after specific amino acids, specific kinds of amino acids. Did you do problems like this on the exam? If I gave you a peptide and said, where's chymotrypsin going to cleave it, or trypsin? Okay. Last slide. Chemical properties of proteins, protein denaturation. So proteins can unravel. This was literally the bane of my existence in grad school. I did a lot of protein research, and you had to purify you had to grow and make and purify your own protein, and then you had to try to study it. But if you don't get enough protein, you can't study it. And if proteins denature, you can't study them. Okay, so this can happen though. What causes proteins to denature or unravel? Well, what it is first, denaturation is a change in the structure at the secondary, tertiary, quaternary level, but not the primary level. So it doesn't just clip it. Denaturation doesn't clip it apart like a protease. It just unravels the wadded up, bound up structure, okay, into its primary structure. So this can happen when non-covalent interactions are disrupted. Okay, so what can disrupt our covalent interactions? We're going to get to a few things that can do that. But when they're disrupted, the protein will unravel. So the peptide bonds, the primary structure though, is not damaged in protein denaturation. And this is why we hate it, okay? Because when we lose our protein structure, we lose the functionality of the protein, okay? So this results in a loss of chemical, biological, and physical properties. If it unravels, it's not functional. It's not going to do what it needs to do or what it's supposed to do in the body. And most, not all, but most denaturation is irreversible. Once it unravels, it's not going to fold again. And that's kind of a mystery. Sometimes they can refold. So we call that protein renaturation. And inherently, the protein has all the knowledge that it needs to actually fold correctly, because it does it once, right? When it's made from DNA, messenger RNA, transcription, all that. When you make the protein, you make the sequence, and then it folds. So it has all the inherent knowledge it needs in its primary sequence to fold. But for some reason, when we denature proteins after they've been folded, often we lose their functionality and they don't refold. Lastly, what agents can cause proteins to denature? Okay, so heat is the number one thing. Number 2, large changes in pH. So extreme pH. Proteins are happy at physiological pH, but at extreme pH, really acidic or basic conditions, they'll denature. 3, physical agitation, or mechanical agitation, I should say. Physical, mechanical. Okay, just like physical shaking or damage to it can cause it to unravel. And then lastly, number 4, so if there's a lot of like, how do I want to phrase this? A lot of like salt, if there's like really highly ionic environments where there's a lot of charges. So if there's a lot of salts are ionic compounds dissolved in aqueous solutions. So I should just say high salt concentrations. So if there's a lot of plus charges and negative charges in the solution that the protein's in, those can cause proteins to denature. So, all right, this is a picture of the structure denaturing, and you just kind of see it's unravel. That is protein denaturation. Any questions on that? For sure, know these 4 agents that can cause protein denaturation and what we lose when proteins denature, the functionality. Okay, wow, I'm tired, but we're not done. We're actually getting to kind of the most important thing now. So I want you to go to your email, because I'm about to send an email to you with your exam. And again, I'm sorry, the pictures, the scans, just because my phone is not great, so not the most high quality things here, but hopefully you can still see your exam. Okay, so Cameron, there's yours. Abby, send, there's